Alright, this video is going to cover Unit 5, where we talk about the different types of cell division, specifically the cell cycle, mitosis, and then get into meiosis. And then we talk about some introductory genetic terms and ideas regarding Mendel and his conclusions. This is a summary video, so every slide in the PowerPoint presentation will not be touched on, as this PowerPoint is a compilation of all the Unit 5 notes. So we're going to start with the cell cycle and mitosis. So generally, the definition of the cell cycle is the lifespan of the cell. And it's split into two major parts, interphase and then mitosis. So interphase is when the cell is not dividing, and then mitosis is actually when the cell divides. So we're going to start with interphase. There are three parts or sections of interphase. You have G1, S, and G2. In G1, you have your normal growth and development. This is what takes up most of the lifespan or the cell cycle. It's just existing. It's doing whatever it needs to do. S, this is where we talked about in Unit 4, the DNA replicating. S stands for synthesis, and if you remember, synthesis means to make. So we are making DNA. And then finally, G2 is where we're going to kind of ramp up to divide. You're going to get more growth. You still have some normal functions happening. And then things like your spindle fibers, start those long strings that you will see in mitosis, start to form. So this really is a preparation for dividing. Then we get into the actual dividing. And the dividing is actually split into two techno parts. You have the mitosis, which is only the division of the nucleus, okay? And that is something that a lot of people don't understand or forget, is that mitosis is the nucleus dividing, and specifically the DNA being moved around. And then cytokinesis is when the cell actually divides. That's the dividing of the cytoplasm, and from here, this is the cell that actually splits. So before we get into mitosis, there are different terms for the DNA that I want you to make sure you're familiar with. The first is chromosomes. Chromosomes are the condensed form of DNA. So these are all versions of DNA. So the condensed form is chromosomes, and you're going to see this during mitosis because it's an easy way to have DNA moved around. Then you have chromatin. This is your loose DNA. Kind of looks like spaghetti a lot of times when I draw it, and this is going to be the form that is going to be seen in interphase. So when the cell's not dividing, chromatin is going to exist. It's still compact because DNA is a huge and very long molecule, but it's going to be easier to get into. And then within a chromosome, you have a couple different things. Each half is called a chromatid. It's the same section or same information on both sides, and then the centromere is what connects them. So chromatin, chromatid, and chromosome all sound very similar, but half of a chromosome is a chromatid, and a chromatin is the loose form. So unfortunately, they are very similar. You just got to make sure you know. Chromosome is the compact form during mitosis. Chromatin is the more loose form during interphase. Alright, so now let's dive into mitosis itself. There are four parts. You have, basically, if you remember PMAT, prophase, metaphase, anaphase, telophase, that is the order that the process takes place. And then after telophase, you have cytokinesis. We talk about them as separate things because they are, but telophase and cytokinesis essentially happen at the same time. So, first thing, prophase is by far the longest and most complicated out of all of the parts. The first thing that happens is the DNA goes into chromosomes. So we form our chromosomes because we got to move the DNA around. We break apart the nucleus, and we form these spindle fibers. So these are right here, these little lines, it's kind of hard to tell, that are going to help. Those are kind of the big points. There's other information here, but those are the big points. Metaphase is where we're going to line up all of the chromosomes in the middle. So if you remember meta meaning middle, that will help you. Anaphase, we're actually going to pull and split the chromosomes in half. So then we have each half of a chromosome, which is called a chromatid, go to different sides of the cell. And the way you can help remember this is that Anna, if you think of Anna as a way, we're moving each half of a chromosome away from each other. So we're actually splitting a chromosome. So if a chromosome is X, we're taking half and half, and they're going different directions. It's the same information, just remember it's different copies. And then finally, telophase is basically the reverse of prophase. So reverse prophase. We're going to go back to chromatin. So we go out of the chromatid, which, because remember, we only have half of a DNA or chromosome at this point. So we're going to go back to chromatin, and then we're going to reform the nucleus. And an easy way to tell telophase is that it looks like a cell is about to split. It hasn't split exactly, but it is splitting. So again, PMAT, prophase, we create chromosomes, we break up the nucleus. Metaphase, everything lines up in the middle. Anaphase, we split the chromosomes in half and they are pulled away from each other. And then telophase, we reform chromatin and we reform the nucleus. And then finally, cytokinesis. This is when the cell actually splits. The definition is a division of cytoplasm. And it happens in two different ways based on the type of cell. For an animal cell, basically the center of the cell is just going to combine and twist until it splits in half. For a plant cell, because it has a cell wall, the cell plate, which is kind of the pre-form of a cell wall or the precursor to a cell wall, will start in the middle and then just divide in half. Now, the cell does not want this happening at all times. So you want to make sure that when the cell gets ready to divide, it is ready to divide. And there are checkpoints, or kind of cues within the life cycle of the cell to make sure that it's going to be okay. So the first is at the end of G1, when it's getting ready to divide or to copy its DNA, the cell is going to check itself and make sure that it's okay, like it has the correct size, that it's healthy. And then once the DNA is copied at the end of S, the DNA is going to be checked for mistakes. And at any point in either of these, if something goes wrong, the cell will try to correct it. There can be external things that can help, like growth factors, which will help with cells dividing, especially when someone's going through a growth spurt as a young child or as a teenager. You'll have a lot more growth factors. And then also, a lot of cells will have cell-to-cell contact sensors, and basically if they are growing and then they contact another cell, they'll stop because then they know that they've kind of reached their limit. If everything fails, the cell will go will undergo apoptosis, which basically means the cell will kind of essentially self-destruct because there's something that's so screwy with it, it doesn't want to cause a problem. That problem being cancer. Cancer is essentially uncontrolled cell growth. That is the definition of the disease. And what you will get when you get cancer is a tumor. And there are two types. You have benign and malignant. Benign means it just grows in one spot. This is the better one if you want to say that. A lot of times this can be just removed with surgery. And then malignant is when it has what we call metastasize and spread. Typically, as this picture shows on the bottom, it gets into the bloodstream. There are several different causes for cancer and treatments, and so on. Alright, so now let's move into meiosis and kind of the differences between them. I'm going to briefly touch on some of the high points of meiosis process, but the big thing for this section of Unit 5 is really the differences between mitosis and meiosis. And that starts with a difference between the types of reproduction. So we have asexual reproduction, where the parent and child are identical. You don't have an egg or sperm. And mitosis is an example of asexual reproduction. You also have something known as binary fission, which is essentially the same idea, but in prokaryotic cells, so things like bacteria. The other side to that is sexual reproduction, where you have an egg and sperm, and the big thing here is that the offspring or the children are different from the parent. You want this in terms of things like evolution. For humans, or for eukaryotic cells, meiosis is what's going to create sexual reproduction. The other thing we talked about here was we have different forms of chromosomes. The big thing here is what we call homologous chromosomes. These are a pair of chromosomes that are the same size and code for the same information. You will always have two of them because you get one from each parent, one for your mom, one for your dad. In humans, we have 23 pairs. The first 22 are what we call autosomes, and the last one, the 23rd pair, we call sex chromosomes. And so, just as a note, we will get more into this in Unit 6. XX is going to be female, XY is male. With chromosomes, we can also talk about the number or copies of the chromosomes. So diploid, di being 2, have 2 copies of every chromosome. So this is going to be our somatic cell, or also known as body cells. So this is all cells except for the egg or sperm. They're all going to be diploid. One thing that's confusing to some students is in humans we have 46 chromosomes. That's our diploid number. That does not mean that if I ask for the diploid number, it should be 92. Diploid, it's 46. So we have 46 chromosomes in a diploid cell. Haploid cell, if you think of hap, sounds like half. We have half the number of chromosomes. So in humans, it's 23. And this is going to be your gametes, which are your sex cells. And the big reason here is that when you put an egg and sperm together, you need to have the correct number. So again, haploid have 1 copy of each. Diploid have 2 copies of each chromosome. All right, meiosis is another type of cell division, but the purpose here is that we're going to make haploid cells. And again, like I mentioned, we reduce the number of chromosomes so that when the egg and sperm combine, we get the correct number. Because if we had 46 and 46 in each egg and sperm, the offspring would have 92, which is not good. And there are 2 major divisions. I say steps here, but it's really divisions. Meiosis 1 is going to be when it divides the first time. You're going to have this homologous chromosome pair that separate by the end of meiosis 1, and you'll get 2 cells. And then in meiosis 2, the chromatids separate. So this process is exactly like mitosis, but you'll get 4 cells. And so we're going to go through these. So for meiosis 1, you have PMAT, which is going to be, and we're just going to label all of those as like prophase 1, anaphase 1, telophase 1, anaphase 1. They all basically have the same thing that happens as in mitosis, but there are a few differences. The big one here in prophase 1 is that you have crossing over. This is where you have sections of DNA that will swap. So if I have my homologous chromosomes, this part and this part will swap parts. And that gives us genetic variability. The other thing too is that your homologous chromosomes pair up. So at the very beginning, your homologous chromosomes will pair up. Metaphase 1, anaphase 1, and telophase 1 are going to be very similar. The only thing here is that in anaphase 1, your chromosome pair, so the homologous chromosomes, are going to separate. Versus the chromosomes split in half. Alright, so here's a picture, and then you can see here, this is where crossing over is taking place, so that's really important. In prophase 1 is the crossing over. And then you can see here, in anaphase 1, you have a full chromosome. It's not going to be half of a chromosome like it is in mitosis. And then meiosis 2, these are all going to be labeled as 2, basically PMAT 2. So prophase 2, metaphase 2, anaphase 2, and telophase 2. The only thing here to really note is that anaphase 2, it's going to be like anaphase in mitosis. The chromosome is split in half. And so this is just showing you, so at the end of the day, you get 4 cells that have 1 copy of every chromosome. Alright, so not going to spend too much time on making eggs and sperm. The big difference here is that you get 1 egg for every division of mitosis or meiosis in females, where you get 4 sperm. That's really the biggest difference, along with a couple other things, but that's the biggest one. This by far is the biggest thing that you really need to know from cell division in Unit 5 is the differences. Mitosis creates genetically identical cells, where meiosis creates unique cells or different cells. You get diploid cells from mitosis. Meiosis, you get haploid cells. Asexual reproduction is associated with mitosis. Meiosis is associated with sexual reproduction. You also get 2 cells in mitosis, where you get 4 cells in meiosis. There are some other differences too, but those are the big ones. And this is just a picture showing. The other thing too that we talked about in the last unit was mutations. So the other category of mutation is chromosomal mutations, and these are mutations that involve the chromosome overall. The main one that I want to focus on, and by far the most important, is going to be non-disjunction. This is where your chromosomes don't completely separate, so either get too many or too little chromosomes. All right, and so what you can see here is down here you get 3 copies of a chromatid and 1 copy there. And so that's called non-disjunction. By far the most well-known disorder with this is Down syndrome. You have 3 copies of the 21st chromosome in Down syndrome. But that is the mutation that really is important. The other ones are important, but that's the one that you should focus on. All right, and then the last topic for Unit 5 was like an introduction to genetics. So in genetics, we look at how things are inherited. And heredity is that idea that things can be passed from parent to child, and genetic is the studying of the patterns. Mendel was probably by far the most important person in genetics to start with. He really was the first person to study it, and he came up with a couple conclusions. Remember that a cross is in the mating of any 2 things, and we have different terms for the generations. We have P generation, which is the first generation, F1, which is their children, and then F2 is the grandchildren. Before we get into Mendel's kind of conclusions, some basic terms. A trait is a characteristic. The gene is the section of DNA that codes for that trait. And then a new term is allele. These are the alternative forms are the different versions of the trait or gene, however you want to describe that. So in the example here, we have flower color is our gene, we have purple flowers, and we have white flowers. Those are your alleles. All right, so Mendel had 3 conclusions. The first was the rule of dominance, basically saying that some alleles are dominant over others. With a dominant allele, you only need 1 allele to show it. We represent it by a capital letter, whereas with recessive, you need both alleles to be recessive to show it, which we represent by a lowercase letter. Generally, a dominant is more common than recessive, but it really depends on the situation. His second conclusion is called the law of segregation. This is dealing with 1 trait. The reason why I say that is the law of segregation and the next one, law of independent assortment, they sound very similar. So this is what this is saying is alleles of 1 trait separate. So you can see in the picture there that if we have a capital T, lowercase t, those are split, and then they'll combine together with something else to make the next generation. But yes, so when you make the egg or sperm, each egg or sperm only gets 1 copy of a trait. The law of independent assortment deals with 2 or more traits. And what this says is that these traits don't influence each other. They are separated independently from each other when we make our egg or sperm. And so your eye color doesn't necessarily dictate your hair color. You can have blue eyes and brown hair. Finally, some more vocab stuff. Homozygous and heterozygous. Homo means same. So with homozygous, they have the same type of allele, and heterozygous is different types of alleles. So in the picture there, homozygous alleles, they're both wrinkled, whereas with a hetero, one's wrinkled, one's round. And then kind of tying everything together, we have 2 different ways we can talk about the genetics. We can talk about the genotype, and the genotype, sounds like genetic or genes. That tells us the genetic makeup. We use letters to represent the genotype. And then the phenotype sounds like physical. Pheno sounds like physical, and that's the physical expression, like what trait you're going to physically see. And tying all those together, we have three options for genotype. Homozygous dominant, where you have two big letters. So in this case, if we're doing the flower color, going back to our example earlier, you'd have two capitals P's. Heterozygous, we have one of each, so we'd have a capital P and a lowercase p. And then homozygous recessive is two little p's. So again, homo means the same. So dominant we represent with capitals, so it's two capitals. Recessive is lowercase, so two lowercase. And then hetero is different, so you have one capital, one lowercase. If you have at least one capital allele, which is your dominant allele, you will show the dominant trait. So homozygous dominant and heterozygous both show the dominant trait, which here is purple. The recessive trait would be white, and you only get that if you have two recessive alleles. Any situation or any problem that you come across or question would tell you what trait is dominant, which one's recessive. All right, so that covered Unit 5. We talked about the cell cycle and mitosis. We compared that to meiosis, and then finished up with some introductory genetic terms and ideas of Mendel.